Good morning, everyone. I am Maria Guarino, MD, PhD, and I am going to present you the preliminary data of this project I developed with Professor Dufour at the University of Bern in the hepatology lab. And the background of this project is to evaluate the impact of exercise on an advanced fibrotic stage of NASH and liver carcinogenesis. The background of the project is the fact that for this kind of disease, actually the only approved treatment is lifestyle intervention with modification on diet and with exercise. So if it is well known that the exercise is able to prevent the disease and to have an impact on the liver steatosis, less is known about the impact of exercise once the disease is established, and if the exercise can be able to prevent the progression of the disease in terms of fibrosis and liver carcinogenesis. So we created an animal model of NASH with the choline-deficient IFA diet. This kind of diet is able to reproduce the phenotype of the disease with the fibrosis and liver carcinogenesis, even if in a context that is a little bit different from the reality of the disease, because we don't have the metabolic alteration that are usually part of the disease. So we don't have obesity and insulin resistance. So we had 4 groups of mice, Black 6N, the group of mice under standard diet as control group, and then we have 3 groups of mice under this special diet. One group have the diet for 12 weeks and then euthanized to have an assessment of the disease before running the experimental part of the project. So we have then further 2 groups of mice, one continuing with further 8 weeks of diet and sedentary lifestyle, and another group continuing for further 8 weeks and exercise, treadmill exercise in particular, 1 hour per day per 5 days per week of treadmill exercise. And after 20 weeks of experiment, we euthanized all the mice. And these are the preliminary results of this huge project. In particular, when we go to compare the sedentary mice with the exercise mice in terms of the biochemical plasma evaluation, we have a reduction of the plasmatic level of transaminases, of bilirubin, of triglyceride, cholesterol, and bile acid. Then we have also a reduction in the liver triglycerides. As you can see in the figure, we have a reduction in the liver triglycerides in the exercise group, while in the sedentary group the amount of triglyceride in the liver is hugely increased. Then we have also an impact of exercise on the histology of the disease. As you can see in the H&E, we have a reduction of the steatosis in the exercise group as confirmed with the liver triglyceride quantification, but the most important result is in terms of the ballooning degeneration, because in the exercise group we have really small amount of ballooning degeneration in comparison with the sedentary group. So it means that exercise is really able to prevent the progression of the disease. And we have also a stability of the disease in terms of the NASH score. Then, in terms of fibrosis, we assess the fibrotic stage with the serious red staining, and as you can see in the picture, going to quantificate the fibrosis in the 2 groups of mice, we can clearly see that with the exercise we are able to stop the progression of the disease, because the amount of fibrosis is comparable between the NASH baseline group and the exercise group, while in the sedentary group we have a huge increase of the fibrosis. So then we also evaluated the impact of exercise on carcinogenesis, and as you can see, the exercise toward the activation on the AMPK pathway and the consequent inhibition of mTORC is able to reduce the development of hepatocellular adenoma significantly. So we have in the sedentary group 100% of the mice developed hepatocellular adenoma, while in the exercise group only 70% of the mice developed adenoma. And we have also different shape of the disease, because in the exercise group, the number of nodules developed in the mice is more or less below 5 nodules per mouse, while in the sedentary group, the number of lesions developed is always above, is more or less above 5. Then we had an evaluation of the impact of exercise on liver sinusoids. In particular, we analyzed at scanning electron microscopy the sinusoids, and as you can see in the control group, the sinusoids are okay with the defenestration, while once the NASH phenotype is established, the defenestration is completely disappeared, and we have a capillarization of the sinusoids. And unfortunately, in this setting, with exercise, it seems we are not able to revert or to ameliorate the capillarization of the sinusoids. Then we also analyzed the mitochondrial bioenergetic respiration with the Ouroboros respirometry. And also in this setting, you can clearly see that the maximum and the complex force-driven respiration is reduced in the NASH group, and with exercise we are not able to restore the usual pattern of respiration in the mitochondria. So what we can conclude is that the exercise in a NASH animal model is able to improve biochemical and histological parameters and can stop the progression of fibrosis and liver carcinogenesis.